So today we're going to talk about protein interaction predictions. By the way, the next lecture will be on the 10th of January. I'm not entirely sure that Jana Schmidt is going to talk. Alternatively, it will be I talking about the next part of this. So protein-protein interactions, there are different words out there, and I want to just familiarize you with the words and to explain and define what we want to do here. What you see here is a complex between a protein called the glycoprotein 120. It's called 120 because it has a weight, it has 1,200 residues roughly, from HIV, and two antibodies, two different proteins, and CD4. Now this is very, very important structure, was published by the group of Wayne Hendrickson in '98. It was important because this has to do with the way the virus goes into the cell, the HIV virus. And essentially the attack, studying the attack is what they hoped, or what they needed the structure for. There are two different versions of this. This is one, and this is sort of a space-filling model where every single residue really is a sphere. But it's the same thing. Here you see in different colors, however, here the two different antibodies here, the CD4 in light blue and the GP120 in dark blue. Now, there are lots of interactions here, lots of interactions that you could call a protein interaction. You could say, well, the way these particles bind, or the residues bind here, is a protein interaction. It's an interaction of two residues, two parts inside of a protein, right? And you could say, well, a protein interaction is something that happens between two different proteins. And shown in red circle here. Or you could say it is something that happens in this particular case for CD4. The yellow circle indicates that it's the same part. This is the same protein, the blue one here, but it's essentially two different domains, two different regions, right? And the way domains interact could be somehow similar to the way different proteins interact, okay? Now, most publications, and it's difficult to count that, but at some point I counted, just going through an issue of a journal, and then the number most was in the ballpark of 95%. 95% of the people, when they talk about protein interaction, means something else. What they mean is a chain of events. A activates B, activates C, activates D. And most people would say that this is a protein interaction, so that in fact A and D interact. They interact because they are part of the same pathway. They interact because all four have to be there to perform a certain thing. So this is a biological process. You need all four to finally get to some end product. And by that force, all these four somehow interact. They contribute to a path, to a pathway, to a role. They have the same sort of role in some sense. For us, for this lecture here, protein interaction essentially really means physical interaction, so meaning that when two things really touch, okay? Now, in particular this one and this one, they do not interact. Again, most people who use the term physical protein interaction actually mean that they all interact because at some time point, all four of those form one complex, one stable unit. And again, there's an experimental way of sort of fishing out these stable units and then saying that everything that is sort of cluttered together is one big complex, is one set of protein-protein interactions. For us, interaction means binding. One experimental way, in fact the most important high-throughput experimental way, is by something that's called yeast-2 hybrid. In the yeast-2 hybrid, so you want to test whether X and Y interact. And you actually put, genetically engineering, put another domain with a reporter, I'll get to that in a minute, on X, and another domain with another reporter onto Y. Now, the moment they interact, you sort of activate an upstream specific activation event that leads to an emission of light. These two interact, you see light. So you see light, you can actually really look at the cell, then you know these two interacted. And that people can really do in high throughput, meaning they can do that for 1,000 proteins at a time. Or for larger sets even. There are many mistakes happening, so you have a lot of false positives, many things you miss. So when the first large-scale used to hybrid studies were published, both on a bacterium called E. coli, both of those, they were sort of published within a relatively short period of time. E. coli has 3,000 proteins, and both published 1,500 interactions. So this was a remarkable overlap and a remarkable event. But the actual overlap was 0. So they had 1,500, they had 1,500, but they had not a single interaction in common. Okay? That gives you some idea about the number of things that they miss. It does not give you an idea about how accurate the ones that you found were, but both have mistakes. But still, this is the best method out there. And there's some groups that do it very well, and that brings us to a lot of the data we have today. Just going back to the example that I showed, E. coli having 3,000 proteins, easy for you to do the math, right? It's the same story that we had on Tuesday. 3,000 by 3,000 minus 1 is 3,000. Half by 1,500 is a rather big number, over 4.5 million. And out of these 4.5 million, so the 1,500 is 1,000-fold smaller, right? More than 1,000 or 3,000-fold smaller. So that you, in fact, we do not know, in fact, what's the average number of protein interacts. So what do we expect? Do we expect for 3,000 genes 30,000 interactions? Do we expect 100,000 interactions? Do we expect 10,000? So at this point, people believe it's more than 30,000, but we don't quite know. But even in a space of 30,000, to have two sets of 1,500 that don't overlap is not that unlikely to happen, right? Anyway, so that is one of the high throughput methods, and in fact, one of the few ones that really tags physical interactions. I have talked again and again about homology-based inference. You guys ready to explain what that is, right? And homology-based inference, we can do the same thing for protein interactions, okay? This is slightly more complicated. So what in this particular case, usually there's a protein A for which I know something, there's a protein A prime for which I don't know the function. A and A prime have similar sequences. I say A prime has the same function as A because they're similar in sequence. Now, the thing is a little bit more complex because what I actually want to talk about is A binding B. So I need a protein A prime, but similar to A, and a protein B prime. So I need a pair that is similar to the ones up there, right? So A prime, B prime, if they are similar enough, then are they going to interact is the question. And now there are in fact different ways in which you can do that. You can sort of do an average over these two. And now what I'm going to show you is data for the similarity of the smallest is larger than some threshold. So meaning that whatever I look at is such that the one of these pairs here who is more distant in sequence, so if A prime is less similar to A than B prime is to B, then that would be the less similar one, and that determines the cutoff. And I'm going to show you a graph with different cutoffs. So these different cutoffs here are from the HSSP line, and so this particular number here is this light blue line here, where I have number of residues aligned, sequence identity, and so the longer the alignment length for 100, the 0 line is blue here, I talked about that, but so the longer the alignment length, the less sequence identity, the more sequence identity, the less sequence identity you need. So this is lower here than there. This point is this point, and so forth, and so on. So this measures essentially distances from this blue. Positive numbers up there, and I only show positive numbers. So this axis shows essentially the percentage for which this is right, but this is misleading. Doesn't really matter. What does matter is that there is some sort of drop, okay? And the drop is such that it seems, given the error bars, there are different ways in which you most likely the function is something like that. And if you put in a function like that, then you would see it works a little bit better for high similarity, and it sort of drops for lower. But the difference is not that high, it seems, on this set. Now we get into the discussion we had before, the word homology. This is not the type of word I believe it's good for you to be familiar with it. I'm not entirely sure that this is the right thing for examens to ask, but I still want to make this idea. The word homology originally comes from the observation that when you look at organisms, you see similarities. This is going essentially back to Darwin. So we see the evolution of species. So we see, for instance, the hand in 4 different creatures here, 4 different species, and in many ways, so they are remarkably similar. And I'd argue the most remarkable similarity is this one here, because whales don't really walk around with feet. Yes?
Why isn't the bird more remarkable?
Yes. You are right. The bird is more remarkable, but actually has 3. It's true. Point taken. So evolutionarily, this is much further removed. The point is, when you see a bird, can you point me to the 4 hands or the 2 arms and the 2 feet, so to speak, of a whale?
Don't whales have fins? Or the flaps?
Those are the ones.
Yeah.
4. They don't have 4 of those. Exactly. That's where the problem lies.
What about the tail fin?
Good.
That should be 2.
No.
It is not?
Wait a minute. I'm not sure myself now. Where's number 4? It could be. My intuition currently is, I have to get back to you on that. Well, we both do Wikipedia on that. But anyway, so homology is essentially something that is done like in a different organism. Homology is the similarity between different organisms. That in principle is the definition. Now, we use this term very, very often for the level of genomes. And so now we get to the 2 new words. I already introduced them again. I will not ask this in the exam: orthologs and paralogs. So we begin here. Say I had a genome of just 3 genes. Again, you all know by now the minimal genome is about 500. Bacteria typically have a few thousand, ballpark 3,000 something. So this is totally unrealistic. But now we sort of have a speciation event, so we form 2 new species. We have another speciation event, and upon this speciation event here from B to C, actually what happens is I duplicate an entire gene. When I duplicate this gene, so this is the duplicated version, the idea is I already have this square gene here. The square gene does what the square gene has to do. Now I have 2 versions of the square gene, and if I wait long enough, then it will evolve slightly into doing something else. Okay, sometimes it may be an advantage to have another square gene around. And sometimes it may be an advantage to actually use something like this square gene and do a new function with it. Because the square gene is already there, so you do something new, okay? And this is a way in which bacteria in particular, but also eukaryotes, acquire functions, new functions. It's a very, very frequent event. So now, upon this duplication, you essentially would have something that you, in terms of sequence similarity, would look like a square gene. But it may have altered its function. And that is what people refer to as a paralog. Paralogs are essentially, they happen through a duplication event, and they are thought to do something different. Okay. Now, have you... If somebody came on April 1st with a jumping gene to you, then you would say, so jumping genes, right? So they jump from here to there.
Okay.
You say, okay. So I have seen the jumping gene. Have you ever seen, or let me invent a way. So have you ever seen corn has different colors, right? When you look at a corn cob, sometimes you see different colors. Not so much in Germany, but in the US. And this is because you have the jumping gene.
Okay. That is weird.
You buy that?
I don't know. I'm skeptical.
Anybody not skeptical? No, everything can happen in biology, right? So McClintock discovered that really for corn, and there really is a jumping gene, and in fact in corn it was discovered, but the jumping gene here is another example for the sea slug. The jumping gene is something that happens all the time in the world of bacteria, but also for us. Our genome is full of things that obviously jumped into it as virus, so it's full of old viruses, but it's also, we all also have traces of things that happen through lateral gene transfer. Is the sort of horizontal gene transfer is the way it's often coined in the literature. Essentially, this is jumping genes, and they can jump as entire sets of genes, and they can jump as single genes. Okay. Now, let's get back to the story of the homology-based inference and make it slightly more complicated. So what I showed you is the data for A prime and A, B prime and B. Now let's assume we have another set of homologs, or similar proteins, A2 prime, B2 prime. But A prime and B prime are in the same species, and A2 prime and B prime are in different species than A and B. Okay? So the traditional understanding would be these are the paralogs and these are the orthologs. So these are the pair of proteins that does the same thing in a different species. That's the idea, the sort of simple idea. And as this is biology, there's always an exception. But again, this is the graph for the orthologs, so in a different species. And this is the graph for the paralogs. So this is what we would have expected to be completely the other way around. So we would have expected that the green is above the blue. We would have expected that if I have the same pair of proteins in another species, they are more likely to interact, because the other species essentially copies the essential process of life. Okay, we all do something different between different species, but many processes are very similar, at least between very similar species. Us and monkey, us and mice even. And the ones that are duplicated adopt new functions, so they would change. But what we see here is just the opposite. And we saw that for a variety of species. We published this a few years ago, or the year 2006, so 10 years ago by now, and it's still not clear. So there's now, others have published similar findings in different contexts. Why that is the case is still not clear. So one simple answer could be, well, keeping these paralogs, letting them do something else, but still having them around if something goes wrong with the original gene as a backup. Maybe that's such a big advantage, but this is still true. We don't know. Okay, can we predict it de novo? So can we predict it just from sequence, protein-protein interaction? And the first thing that I ever did is I essentially had a stretch of residues from protein 1, a stretch of residues from protein 2, and I wanted to predict, in this particular case here, in the window of 6 residues on either side, where's the interaction? Okay? And I trained a machine learning on that in '98, and it totally failed. It absolutely failed. And ultimately, the problem is too many false positives. And so I got into a long journey of removing false positives. So one, in fact, would be to predict surface residues. So what I mean here is when you look, say there's the core, there's the solvent, and the light green here is the surface. Just like for subcellular localization, I told you that the environment is imprinted upon the surface. Proteins that interact on the surface. Okay? Now, if I could predict the light green here, then I could reduce the false positives, and in fact, not only slightly, but since sort of half of the residues are on the surface, I cut my mistakes by a factor of 4, right? I keep is 1/2 times 1/2 is 1/4, so I throw away 3/4. Okay, in what I throw away, there will be some things that are wrong, but most of what I throw away will be false positives, right? And we went through a couple of methods that did something like that. And so we predicted residues in external interfaces separated internally. We predicted subcellular localization. And so that I already showed you. We predicted residues in protein substrate interfaces. Some of that is still going on. We predicted domains, we improved alignments, and then Tobias put it all together. And I'm going to talk next year about exactly what he does. But before I talk about that, let's talk about the method that predicts, in fact, the interfaces to other proteins. For the interface to other proteins, again, what we want to predict is that these two proteins bind, and I want to predict this now from sequence alone. So I do not know that GP120 binds CD4 here, or the antibody. I just have the sequence of GP120 and want to say, here are the sites that bind another protein. What the other protein is, I don't know. Can I say that? So, and for that, in fact, the question becomes, on this slide here, I show a bunch of different types of interfaces. So in this green one here, this is an interface between two residues inside of a protein. Okay? Then there are sort of external homomers here. So you have the same thing that is forming a dimer. So there are two copies of the same thing, they bind each other. And then there are transient protein interactions. Transient, what it means is they go on and off. So the protein is moving around in the cell, and then it binds the other one. So this one is outside the cell, it comes outside, and then the binding is happening. So in that sense, it's transient. At some time of the life of the protein, it is there, and sometimes of the life of the protein is not there. Right? That's the definition of transient. Permanent heteromers are simply two different parts of the same protein. And the first question here really is, are they the same or are they different? Because if they all would be the same, if there is something such as the biophysics of a binding interface, then in order to learn these, all I need to do is predict binding sites. And binding sites I can easily predict. PDB, the database of proteins with known structures, is huge. I could take the entire PDB and predict binding sites, if that would be the way to do it. So how do you find out whether it's actually true, whether they differ or not? Let's talk about some. The answer is on a social level more than on a scientific question. What do you do? So the person who did that was Yanai Ofran. His first step was what? So that was the question we asked. So we want to develop a machine learning device that predicts these interface residues, and now we're looking for the data. So where do we get the data from? Should we take internal interactions and transient protein interactions into one bag, or not? How do we answer that? So yes, we could try, but ideally we spend, let's say, a few days before we try, because doing that will take longer than a few days. And if that is the wrong start, then we will have wasted more than a few days. So what's the first step? And you do it every day. Back to where's the fourth, where's the foot or whatever hand of the whale? What do we do? Google. We Google, right? We begin by Googling. So you look at the literature. You look at what other people have said. And then what you find is that other people have said they are the same. And yet other people have said they differ. So what do you do then? You read the literature in more detail. And in more detail you see that they actually did different things. And everything that you see, you understand why they did what they did, and you have some criticisms, and I understand why others did it differently. But maybe this whole thing is not right. So are you now ready to believe? What's your next algorithm? You're going to look at, well, the ones who say they are similar, is that a more important journal, right, or more important conference? This is the way we approach this problem. All of this can be done in half an hour. Or the next algorithm would be the ones who said they differ. Do we know them, and do we trust them more? So that's the social aspect of science. It's very, very important. All of that led nowhere. We essentially had to redo the data. How we did that I get to later. This is what we got. These are the 20 amino acids. This is frequency in that particular class, and I'm showing data here only for 3 different classes, versus background. Background is every residue in the Swissprot, in the database of annotated proteins. So above is observed more than expected, below 0 is observed less than expected. Have I shown you that? No. And the 3 different ones, green is internal, Blue is sort of chain, is inside of a protein, but two different units of a protein, so to speak. And red is the transient protein-protein interactions that we actually want to target. What I like now to do is that every single person in the room gives me an observation of what we see on that slide. Because this is what you have to do in your thesis. This is what you have to do for a manuscript. You put the data out and you describe it. That's the results section. Just describe. What it means is part of the discussion, the interpretation. So first you just describe results, and that's what I'd like to do. Nicholas, let's begin with you. What do you see? Everybody observes one, ideally only relatively short statement, and then the next one finds something new.
Okay, let me see if I understand this graph correctly. The Y and the W residual seems to be a very interesting feature.
So W and Y, tryptophan and tyrosine, clearly stand out because for all of them they are much less than expected. Yes. Matthias.
First, I have to ask again about the y-axis.
Fair. Tijana, I'm sorry. You will have to quickly get to scale because soon you're going to answer. So listen to the answers. The y-axis essentially. So I'm essentially having three pots of points, so to speak. The red being transient protein-protein interactions, the green being internal, so pairs of residues that bind internally is green, and blue is pairs of residues from the same protein that bind between different units of the same protein, okay? And then I ask for the blue, red, and green baskets. So each basket contains a pair of residues, but I can sort of take them apart and just ask how many alanines are in this basket, right? How many A's, R's are in the basket, okay? And then I ask, in the green basket, the R's, do they occur at the expected frequency? And the expected frequency, well, I could say 1 by 20. I have 20 amino acids, so I do 1 by 20. But rather than doing 1 by 20, I actually take proteins in SwissProt and use that to compile what's the frequency of all of these 20 residues. And then I simply ask, is that occurring in the green? Is the R occurring in the green more or less often than expected from the background distribution? And the answer for green would be slightly more often for the R, and for the red and the blue and the red, much more than expected. That's what the y-axis means.
So it's really the general distribution of the amino acids in all of the proteins in this protein.
Yes, correct.
So the number to compare with.
Yes.
Okay.
And essentially it's 20 numbers, right? So each amino acid, you only ask how often do you see the R? What's the percentage or fraction of R in SwissProt? In every single protein in SwissProt.
Compared to the pairs in our baskets.
Exactly.
Okay.
So what's the surprise in the basket? The Christmas surprise of the green basket. And again, Nicholas said the one outstanding feature is that W and Y, again, you don't need to know these names, I'm just tryptophan and tyrosine, are really, through all the three baskets, much less than expected.
You could argue that they somewhat, basically you can read it that W and Y, that the amino acids.
No, you have to say something new.
Removed.
No, you have to. No interpretation, just an observation. Matthias. So you're not writing discussion at the moment, the discussion section. You're writing the results section. What do you see?
What do I see?
And it must be a scientific statement. So it cannot be of the type I see red, green, and blue. This is what I will not accept that. Anybody jumping in?
I think for S,
Serene?
yeah, for S,
I got it wrong, yeah.
It's more like...
Oh, you turn Nicholas around.
Yeah.
Yes. S turns Nicholas around. Yes. S is Nicholas -1 or times -1 or something. True. Yep.
I think in general you could say.
You still have to jump in. The longer you wait, the more complicated it gets. Perfect. Yes.
I think in general you can see that mostly half of them are over the average and mostly half of them are below the average.
Which is in fact not trivial. And we see that it's not trivial because they could all differ. And here we see an example where they, I mean, so far the two who said something talked about the ones where they clearly differ, all differ. So that in fact there is sort of a balance between them, as you observed, is not trivial. Two more. Whoever jumps in first.
Only what I see is that in C, for example, the frequency of chain to chain is the highest.
See here, yes, and in fact that is, yes. The most surprising statement of any is the one for cysteine, C, and that is for chain-chain. That's true. And, yeah, anything else? No?
No, give me a chance. No, what's interesting to see that Nicholas mentioned the outliers, those values where everything is below average. But interesting is that there are also amino acids that differ in, so chain.
Yes.
They are below average, but for example, I'm looking at R, for example, for the internal type, they are above average.
Very, very different in internal. And again, so, okay, here's another one.
It's the more...
So, but yes, you see them now here. V, I, L, exactly. Niklas.
That is actually what I wanted to say.
Too bad. So, but ultimately, one thing that you see is that essentially, so Patrick began to count already, but ultimately they all differ, right? So there's not a single case where all three are absolutely identical. Yes, there are cases where they are on the same side, but the length of the bar differs, okay? If we just look at delta, the delta is throughout, yes?
Then the delta is pretty low for, what's G?
G is glycine. Glycine.
Then it's very low for, the delta is very low for glycine.
Yes, that is true.
And it's, I believe, the only...
and I believe, again, this, by the way, is an important statement. But it has to do something with the feature of glycine. Is the sort of most flexible alanine and glycine. The alanine is sort of the most neutral substitution, and there you do see a difference. But glycine, in some sense, is the most flexible substitution, and I believe there's a biological reason for that. But now, having said that they all differ, in some sense, the big question is, are the differences meaningful? And the question to you is, how do I answer this question? So the question is, are the differences meaningful? How do you answer that typically? In fact, there are two sort of, there's a real answer and there's a sort of, if you don't know the real answer, kind of answer. Anybody has anything to jump in? Yeah?
In the past, when we discussed if some statement is meaningful, we always looked at the standard error.
We looked at statistically significant. That is in fact, if you don't, so meaningful, scientifically meaningful, actually means you have an intuition for it. You have an intuition as a scientist. What is an important difference? What is a meaningful difference? For the amino acid composition, we have absolutely no intuition. This is not something that we can directly observe. It's not something that is part of our intuition building system. Once we leave the intuition building system, we have to get down to the statistics. And statistics essentially is statistically significant. So one way we can test that is chi-square. Now chi-square test, one problem here is if we had small data points. That is not a problem because we have millions of points here. There is a tiny, less well-known problem if the data set is too large, then chi-square also doesn't quite work. This is not, many people may not know that. And that, in fact, we have to address. Anyway, so what we have here, in our set, all differences are 10 minus 300. That was sort of the resolution of the machine that Jan and I used for that in the chi-square test. Okay? So typically, when you have values of 10 minus, I don't know, 5, 3 in biology, or 10, depending on what kind of experiment you do, you publish. So 10 minus 300 is clearly significant. Now, you already know that there is going to be coming something else. Before you publish, what else could you do? To just see that really everything is kosher. Or halal, for that matter. Well, and I had this weird idea of just instead of putting the interaction, the red, green, and blue, just put all the proteins that have a name from that is called A or B in one pot, all the proteins that are called C or D in another, and test what would be the differences between those, 10 minus 300. Then he looked at all the proteins that were published by one author, by two authors, and by many authors, 10 minus 300. Then he looked at random subsets, 10 minus 300. Then we visited the statisticians and they said, well, are your samples statistically independent? Well, you know, Darwin, all the proteins are related. No. So we cannot do statistical tests on biology or what? Whatever the answer is, this one didn't work. That is clear. It's a standard test, and it's the right one to use for this sort of thing, but it didn't work. So how else could we do it? And what we kind of came up with is a well-established statistical significance test. We call it find self, just because it's very intuitive and simple. So you take say this is data set 1, green, red, and blue. And you put all the points in this basket, and now you take randomly from each of these baskets 1,000 points. And now you take from the first one randomly another 1,000. Okay? And then you ask the additional 1,000 you picked up from the first. In a Jensen-Shannon sort of similarity sense, does it match most to this, most to that, most to that? And you repeat this. Random draw, right? You can repeat that 1,000 times. You pick just to confuse you a little bit, I use 2 times 1,000. There's no real reason for that. So I have 1,000 in here, and I have 1,000 repetitions. Okay? And then if we always find this one, if that is always Jensen-Shannon information the closest, then that means that there's something unique about that basket. Okay? And here is the answer. Now the answer is not for 3, but for 6 classes. The internal here are still in green. They changed the color. That used to be the red one here. The hetero-oligomer. So we call them obligomer because this is an interaction between 2 different parts of the same proteins. They're obligatory together because they cannot leave each other, right? They're bound together as one protein. That's why we call it an obligomer. For the homodimers, we simply mean this is the same chain that binds itself. And there's a homo-obligomer. It's just when I see protein A, copy of protein A binding, so whatever. Protein A and a copy of protein A, so it's the same protein binding itself. And I never see this bind to anything else. Then I call it an obligomer. So it's obligatory that they bind because this protein seems to not be bind to anything else. Otherwise, I call it an oligomer. Anyway, so what you clearly see is that the highest numbers are on the diagonal. What you also see is that there are sometimes confusions. Let's get into the domain. Domain is again 2 subunits. So they are confused with internal. Well, somehow understandable. But the most often confused actually with the hetero-obligomer. So hetero-obligomer has 2 units of the same protein. Domain is also 2 units of the same protein. It's just, I call them something else in some sense. So this confusion makes a lot of sense. But overall, I see a clear signal. Now, when I now do random, when I now do authors of 1, 2, 3 authors or proteins starting with A and B, I no longer see anything. I don't show you that here. But this now is a clear test that it works for the real data, but not for random subsets of the real data, for random assignments. Okay, so there's a clear statistical significance. And in principle, we can publish this. But how else could you prove or put one on top? If they really differ, you should be able to predict. If they really differed, then you should be able to, in fact, take sequence alone and predict it from sequence. And that's what we're going to try. Again, here's the interaction that we want to predict, and we use PDB for that. There's a little bit of a problem how to extract the interaction points. There's something about thresholds, and there's something that was important about whether we picture protein interactions. So this is protein 1, and this is protein 2, or B and A. So this is one way in which they could interact, and B and A. This is another way in which they could interact. The difference between the two ways is that in this particular case here, the parts that interact are far apart in sequence, while in this particular case you have sequence similar. So if you cut out a window of 9 consecutive residues, you will see a lot of the interacting residues in there. And that is in fact what this one shows. If I cut out a window of 9, how many interacting residues do I have? I show in yellow data for when I define the 2 residues as interacting if the closest atom is 4 angstrom, there's 0.4 nanometer, and blue if it's 0.6 nanometer or 6 angstrom, right? It's just 2 different thresholds. But you see for both definitions, essentially, you clearly, the sample where there's only 1 residue in a window of 9 interacting with the other protein is an exception. In fact, for most of the interfaces of 9, we see that roughly half of the interface is interacting. So it is not quite that. Then the peak would be more on this side, but much more like this than like that. Okay, that is good because we want to predict from sequence. We want to capture an effect of an interface. I don't know the 3D structure, so I cannot in fact see the binding site. I cannot see what is in 3D similar to each other. I can predict surface, but surface doesn't mean that I know this one is near that one. Okay? Yeah?
Well, I can, at least to a certain extent, predict structure from sequence.
Yes, but so first of all, for about 60% of the residues in human, we cannot do that. Full stop. Secondly, when we did that, the number was much, much smaller. Thirdly, whether that so currently that for many of the 60%, this is not what we get. The resolution of what exactly is the surface patch. For most of the 40% for which we can predict structure, we somehow see something about the core. Sometimes we see binding sites, but so in practice, mostly we can't. Yes, we can, but mostly we can't. And this method should work for everything. Is the idea. It will fail for some things, but it should not be restricted by some availability other than the sequence. Okay. Okay. So brief insertion. No, I'm not going to put in the threshold issue. Machine learning. How will we choose the input features? Now, the social way is you ask somebody in the group, and again, do this. The interactions that you have in the group when you do your thesis are very, very valuable. Now, sometimes the people you ask will feel that this person is asking too much. So try to find a way, again, a social way, to make that person happy. And that may be that, you know, You pay for the coffee, or every morning you bring your coffee. Or there are different ways in which you can do that. Pay something for your question. I mean, be nice, but do more than that, because giving advice is something that is, the social interface is extremely important. Science is communication, and a lot you can avoid. Don't ask one person, ask several people in the group. Now, this may or may not work. So in this particular case, they may, in fact, no matter how nice they are, they may argue, well, I actually don't know, because what you work on is a new problem. For this particular case, when Jan and I worked on that, we didn't know. So how do you do it then? Well, what I would do is I would sort of ask my intuition, which typically is actually my memory, and then would take what we did before. But since it's a new problem, this may or may not work. Because I have a lot of experience, most often it will work, but for real new discoveries it will not work. So what else can you do? What people do, and Matthias, you said that you have done machine learning before, what people do is they take a bunch of features and see a single feature, test a single feature. How well is that working? And do that for all the features, and then they pick the one that works best. And then they add one. In practice, this very often goes wrong. So it is usually better to somehow see, is there anybody who has done anything that is somehow related, and use those features. And begin to somehow try with a bunch. Don't work it up from one feature at a time, but try with a bunch of features. Bunch could be 50, could be 100, depending on your problem. Okay? Now, the simplest feature could be what? So again, what we want to predict here is these residues that are in the binding site. I will not teach the method, the interaction. I will only teach, so my training set will only contain these black dots, saying the black dots are the ones in transient protein interactions, and the one, all the residues without the black are not. And that's what the system is supposed to learn. What's the input? Simplest, what would you start with?
The properties that usually need to bound.
Yes, that is not essentially, not exactly the simplest. Well, you have to learn the word chemical for that. So yeah, fair enough. I mean, the simplest thing is really amino acids, right?
Yeah.
So if you, but now, remember how we did it in subcellular localization? Do we? What is the input for predicting subcellular localization? So Tanya's way is k-mers. But what's the simplest way of, in fact, taking the entire protein and predict whether it's nuclear? What's the simplest input for that?
We had the amino acid composition.
20 units, exactly. Can we do that here?
In every instance is an amino acid itself. Did I get it correct? It's basically...
So this is an amino acid, this is an, so every dot is an amino acid, yes?
And this is our, so to speak, one row of our data set, right?
I'm not entirely sure whether I want to answer your question, because ultimately you're asking the same question that I'm asking. Ultimately, if I answered your question, then I answered my own. So I cannot answer your question because I want an answer from you, yes?
So to take this correctly, we take a number of amino acids from protein A, number of amino acids from protein B. You want to see if they bind.
No, so I'm sorry, let me... No, that I did not explain it clearly. So we, yes, A binds B, but I remove this. Oh, good. Yeah, you need some more fantasy. Christmas cheer. All that you will see is A and the black labels. And you will not see a structure, you will only see the sequence. You will not, I don't give you B.
Yeah, but I have two proteins and
no.
One protein.
All that you will see is protein A.
And I want to get the bindings.
And I give you the black dots. I mark the black ones. So exactly, I tell you, these are the binding sites, residues.
Does a protein always have the same binding sites, or it
No.
depends on what kind of protein?
So it doesn't, yes, so the number of binding site residues differs between 10 and 80 or 100 almost. So this already is very variable, and that depends on the pair in which they bind. There are different types of protein-protein
We
interaction.
don't know B, but we have to find out which amino acids from A will be the ones that bind.
Exactly. And my hope is, so let me go one step back to motivate why I want to do that. My hope is that my problem doing A against B, so sliding it through, trying all possibilities between A and B, I had too many false positives. The hope is that if I can predict the binding site, then I don't have to slide the entire thing. I have, in sliding A and B, all I need to do is focus, zoom into the black ones here, and then ask, I hope that I can have some black ones in B, and some black ones in A, and now if I want to predict whether A and B bind, all I will try is the black ones. And since the black ones is a tiny subset, or like 10 to 20% subset of the entire residues of A, say it's a 20% subset, then now surface versus non-surface, I reduce the problem by 1/2 times 1/2. Is a quarter, so I reduced it by a factor of 4. In this particular case, just a moment. Let me continue my statement here. In this particular case, by getting the right black ones, I would reduce the false positives by a factor of 25. So this is why this is such an interesting method for me, because it's going to make a huge jump. So instead of looking at all against all, I have to look at 1/5 against 1/5. And that is a factor of 25 reduction. Nicholas.
We saw the graph before, the one that we discussed.
Yeah.
And there we saw that there are proteins that are very likely or usually bind protein to protein. Mm-hmm. So we could just take the input set of the number of proteins. We take as input set a vector of 20 amino acids. We know how likely are they to be binding to protein, so the red label. Mm-hmm. And then we go from there. So we know that, for example...
But that is, so think it through. So that will essentially be one input per protein. Right?
No. We take the black dots. We count for each amino acid.
Yeah.
We count each amino acid.
Yeah.
And then we know how many of each amino acid are there.
Also, you give essentially a probability for each amino acid, and then you assign a probability. So essentially, you look up for each black dot. You ask what's the probability for that. So then the input would actually be 20, and it would not represent the 20 composition, but it would represent one residue at a time. And then you move around in the entire protein. Is that what you're suggesting?
No, actually not. What I want to know is...
But wait. I still want an answer to my question.
Simpler, actually.
Simpler?
Yeah.
Oh.
I just take this black sequence of black dots. Mm-hmm. We have a category for binds with protein, so protein, binds within protein, and so on.
Oh.
We can see, are there many amino acids that are in the category of protein-protein binding? And if we have like a vast amount of them in our black sequence, then this should be a sequence that is a binding site. Or not.
And our lecture ends.
Really?
I'll get back to that. Let me just ignore what you say for a moment. I'll get back to that. Within the next 15 minutes, you will understand why I ignored it. But let me just show you one simple way in which we can do it. In the simple way in which we do it, what I show here is a window of 13 consecutive residues. Okay? So we take 13 residues. This is the central, so that's position 1, 2, 3, 4, 5, 6, 7. And my system is supposed to learn whether the 7th residue is in a protein-protein interaction or not. Okay? Now, each one of these blurbs here essentially represents all the amino acids, so essentially represents 20 input units. Not quite. I need more than 20. Why? Because I want to move this window. Essentially what I want to do is move this window through the protein, right? And in moving this window through the protein, this central residue should at some point be number 1. The first residue in a protein. I want to predict whether the first residue in a protein is in a protein-protein interaction. If I want to do that, this is residue number 0, -1, -2, -3. And they're not amino acids. I mean, there is no residue before the first, right? So I have to introduce a unit, the 21st here, that codes for, let's call it the spacer. So the thing that is not an amino acid. Alternatively, one way of coding this here, the simplest way of coding that, I have 20 amino acids, and at every single point I observe 1 amino acid. So that unit will have 19 zeros and 1 1. If there's a P, then the P unit is on. If it is before the spacer unit, then the spacer unit is on, right? That's the simplest way of coding it. And then, you know, there's a strongly predicted, a weakly predicted, both are predicted because they both are higher. Here in this unit, both are predicted to be protein-protein interactions. I can use more information, let's not go into that. The most important information that I use, so essentially what this one says, you use predicted solvent accessibility, I use predicted secondary structure, and I put this into a combination of machine learning devices. But that is very important, but the most important thing really is a profile. And the profile, what I do is now I read the protein sequence in this direction. Here's an alignment. So this is my cutout window. Here's my central proline. For the central proline, I see that all proteins that are similar also have a proline. They all have a D, have a G. Here, for this D, some of them have a V and some have an E. So in all of these cases here, PDG is trivial as before. So the PDG unit is on and everything else is off. In this particular case, the D unit is on to 20%. The V unit is on to 40% and the E unit is on to 40% too. So essentially now, within this blurb here of 21 units, in this particular case here, where I see V, V, E, E, D, I will have 3 lights on that together carry 100%, so 40, 40, 20, right? And so forth and so on. So again, I use the same sort of system, the same input as before, where we use just amino acid sequence and put the profile in, and that is immensely important. And the other important thing is really to use secondary structure, solvent accessibility, membrane region, and disorder as input. And I believe this is where we stop it.